Right, this lesson is about how enzymes work as catalysts. Hopefully by now you understand what both of those terms are. Enzymes are a certain group of proteins. They're very important in living organisms. And catalysts are also incredibly important for all of the chemical reactions that need to happen. Okay, so our guiding question is, how do enzymes work as catalysts? And this means that you should also have some guided notes out in front of you. And be taking notes, filling in the blanks, whatever you have in front of you. All right, let's get into it. So catalyst is something that speeds up a reaction by lowering the activation energy. Now, if we were to look at this mathematically, we could plot out the energy amount in the y-axis and time on the x-axis. And this means that at some point you have a certain amount of energy, and moving forward in time, in order for a reaction to happen, you have to add some energy to it, and afterwards it becomes lower in energy, which means it's more stable. Okay, this gets into some chemistry and physics that we don't really need to cover here. But anyway, this is what it looks like mathematically. And when catalysts are present, less energy is needed for this reaction to happen. Okay, so without a catalyst, this much energy would be required. With a catalyst, with an enzyme, only this amount would be needed. Let's put it in more understandable terms. Okay, so a catalyst is a sort of matchmaker. Okay, let's say this lady here knows this guy, and she also knows this guy, and she thinks, you know what, they have some shared common interest. I bet I should introduce them to each other. So she says, hey Mike, you should meet my friend Bob, or Bill, or whatever his name is. Let's go with Bob. And so she takes Mike and Bill, forgot what his name is already, and she says, hey, you should have lunch together. She is the catalyst that brings the two of them together. Okay, otherwise this guy might not have ever run into his new friend, so she has sped up that reaction. Okay, catalysts do this in the body. They take one chemical and find the other chemical that they want to put it together with, and therefore they speed up the reaction. So we could not do all of the things like cellular respiration and pumping things through our hearts, and all of the different reactions that happen in our bodies would not be possible without catalysts and these enzymes. Okay, now enzymes can bring molecules together like that matchmaker did. So this would be what we would call a synthesis. Okay, they would bring this triangle together with this, I don't know, not exactly a square. What is some polygon, right? And they make this. Or another enzyme might bind to this and break them apart. Okay, this is what we'd call digestion and synthesis. But each of the enzymes has one job and one job only. That's very important. Okay. Now, as catalysts, enzymes speed up the chemical reaction. Okay, this is the cycling of matter, right? We're moving things around in our bodies. This is constantly happening in our cells. Okay, for example, the Krebs cycle shown here is very highly controlled by all of these different enzymes. Citrate synthase, you don't have to remember this. Aconitase, I should be able to, I don't know if I said that right. Isocitrate dehydrogenase, okay, alpha ketoglutarate dehydrogenase. All of these are enzymes. All of these guys in teal are enzymes. They are doing one job and one job only. Okay, fumarase takes the fumarate, adds H2O and sends it and makes it into malate. Malate dehydrogenase takes the malate, grabs an NAD plus, changes that to NADH. Okay, you see the picture here. Okay, each of these enzymes is involved in this part of the Krebs cycle. Okay, this is a very important cycle in cellular respiration. So every single chemical reaction that happens in our body is controlled by a protein are called an enzyme. Okay? You don't have to remember this. Not yet. Maybe in college. Maybe if you take AP Bio. Okay, so let's say you wanted to break down this brick wall, or you had it between your neighbor's house and yours, and you kind of like your neighbors and you think we should hang out more, but we have this brick wall between us. You could wait for a really long time, or you could wait for a hurricane to knock it down or somebody to drive their car through it, but that would take a long time, right? That would be the amount of time that it would take to crumble on its own. If you had an enzyme, you could break down the wall. Okay? In this case, our enzyme, I would say, is more like a sledgehammer. Okay? If you took a sledgehammer to it, you could probably be hanging out and having bonfires with that neighbor within the weekend, right? So enzymes are kind of like the sledgehammer in this case, and they can break down this brick wall. Now, if we took a sledgehammer, because we didn't like this blanket for whatever reason, we couldn't really use a sledgehammer to break this blanket apart. It's made of different materials, right? It's made out of yarn, made out of granny squares. This also could fall apart on its own over time. Blankets happen to do that. But you could also take a pair of scissors and cut through the middle, right? It's a very beautiful blanket. I wouldn't do that, but to each his own. Okay, so you couldn't use the scissors to break down the brick wall any more than you could use the sledgehammer to break down the blanket. Okay, in the same way, different enzymes are specific to their substrate. Okay, if they need to break down carbohydrates, they break down carbohydrates, and probably specific carbohydrates at that. If they break down lactose, they're not going to be able to break down glucose, right? So the enzymes are specific to their substrate. Important diagram here is this enzyme substrate complex, and how we take an enzyme and a substrate and make an enzyme substrate complex when the two come together. Okay, this active site is where the substrate can connect. Okay, the shape of the active site is incredibly important, or it won't bind to the substrate. Okay, now after this enzyme looks to be one of the ones that's breaking things apart. Okay, so we have our reactants here. These are the things that are reacting, and these are what is produced. Okay, we call those the products. Okay, so some really important terminology here is the enzyme. Okay, that is the protein that breaks or combines the substrate. Now, importantly, the enzyme does not change. Okay, the substrate is changed. In the process, the enzyme is not changed. Look, it's the same shape. It's going to go off and do the same thing to another substrate. The substrate is the stuff that's being broken down or synthesized, right? That's what's reacting. The reactants, okay, in this case, this is the reactant. This is what goes into the reaction. It's also the substrate. Okay, lots of overlap here. The products are what comes out of the reaction. The enzyme substrate complex is the complex of the enzyme and the substrate together. It sounds very complicated, but the complex is two things together, okay? And in this case, it's the enzyme and the substrate. Okay. All right, please do not forget this. Enzymes are proteins, okay, which means they are made up of a long chain of amino acids. The amino acids are the monomers, right, that go into making the proteins. Now, the proteins start off as this long chain of amino acids, and eventually, after they're formed, they will form into a 3D structure. Okay, this is lactase. This is the enzyme that breaks down lactose. It is a long strand of amino acids, but it's made into a 3D structure, kind of like a big blob, but that forms a particular shape that can connect with lactose and break it down. Okay, the shape of the protein, lactase, is incredibly important. So those are some really important things that we need to learn about enzymes. Make sure you understand the words, right? Products, reactants, substrate, which one is the enzyme, what kind of macromolecule is the enzyme, okay? What is the complex called when the two come together? Stuff like that. All right, thanks for listening.